Good morning, dear students. Today we are going to start with a new topic that is bioinformatics. This particular topic was not there in your syllabus in first year and second year. Basically, bioinformatics is the science which is consist of both biology and information technology. Okay, so let's introduce bioinformatics in general, and then we'll go in more details about different databases, how the information technology is used in biotechnology, how it is used in biology in general to solve different problems associated with biology. So, introduction to bioinformatics and bioinformatic web resources, that is NCBI, EBI, OMIM, and PubMed. As I said, bioinformatics is a field of study that uses computation to extract knowledge from biological data. There's a lot of information in the form of coded data in the biology. Now you'll ask where this coded data is available. So it is available in the genetics in the form of DNA. Okay. And we know the information about the ATGC. And these nucleotides, when they arrange in a particular manner, they forms a DNA. And this DNA is important for protein synthesis. Okay. So it includes the collection, storage, retrieval, manipulation, and modeling of data for analysis, visualization, or prediction through the development of algorithms and software. So we'll go one by one. So collection of data, we collect data from different sources, okay, about any biological things. Then storage of that data, retrieval whenever it is required, we have to again use it. Okay, retrieval, manipulation means changing that particular data or make some or other changes, manipulations, and modeling of that data for analysis. So finally, only data is not sufficient, you have to analyze the data and you have to come to particular conclusion. Visualization or prediction through the development of algorithms. So here you're going to use algorithms and softwares, which is again related with the mathematics. Okay, so with the help of softwares and mathematical tools, you are going to solve the problems related with biology. That is the core of bioinformatics. It is an interdisciplinary field that develops methods and software tools for understanding and analyzing biological data. It combines computer science, statistics, mathematics, and engineering with emerging new concepts, theories, and techniques in biological analysis. Huge amount of data is being collected by scientists after conducting various experiments. Okay. In, uh, in simple words, if you have a lot of data about DNA, okay, it is very difficult to analyze the data because it may be of hundred pages about genome sequencing. Okay, maybe 200 pages, and manually to go about each and every nucleotide and analyze it in the form of coding is very difficult for a human brain. So, with the help of softwares, whatever information we have put in that software, we can easily analyze in a minute about the data which is present in that particular file. Okay. The amount of data grows exponentially. It becomes impartial to analyze them manually. And this is where computer science techniques intervene together with statistics, mathematics, and the engineering. Okay, so our brain has limitations because we cannot store lot of memory in our brains. But in computer, we can store the data, we can analyze it, and we can retrieve it. Whenever it is required, we can retrieve it. Okay, so that is the use of information technology. And whatever study we do in a wet lab, that is whatever experiments we do, whatever data we get from the gene sequencing, that we have to provide to this bioinformatics softwares. And easily these softwares, they will analyze the data and they will provide you the final information. Computational techniques are used to analyze this large amount of data more accurately and effectively or efficiently. Hence, bioinformatics can be considered as a field of data science for showing or solving problems in biology. The field of bioinformatics is considered as silico, where biological experiments are conducted in silicon chip or in the microprocessor. Now you must have seen to how long it took to sequence human genome. It took almost 13 to 14 years. Okay, with many scientists from different countries, they were working on this human genome project. But when this genome sequencing machines like sequencer or this bioinformatics techniques have been introduced, it became very easy for sequencing of any animal. Okay, these days you can make sequencing of any particular animal within a few hours, even, or within 1 or 2 days. Okay. So one major advantage in silico method is that to run simulations and carry out experiments without using any animals or reagents. So this is called as dry lab in a simple words, where we are not doing any experiment. We are just and putting the data in the software and without doing any experiment, we are getting the final result. So, bioinformatics is a combination of subjects such as genetics and genomics. It includes genes, DNA, RNA, proteins, and techniques to discover and analyze various patterns in them. Okay, so they can easily make models, they can easily do the data analysis about the DNA, RNA, proteins, and they can easily see the patterns. Okay. A large data is handled, so it is crucial to have a good understanding in statistics and programming language such as C, C++, Java, R, Python, and Bash. The starting point of curating bioinformatic resources for the public is marked by the establishment of US National Center for Biotechnology Information, that is the NCBI. This term, that is the NCBI, is very important, and it may be asked in the question paper also about information about NCBI, that is National Center for Biotechnology Information, which was established in 1988 in USA. One of its many purpose, the initiative of Human Genome Project. Okay. So, with the help of or within the NCBI, the Human Genome Project was initiated. Okay. As Human Genome Project is prepared to scale up its sequencing operations. About 5 years into the project, a new discipline, bioinformatics, became inevitable. It emerged originally for processing and sharing genome sequences, known as genome informatics. In the first 5 years plan of HGP, it was pointed out that genome informatics is a scientific discipline that encompasses all aspects of genome information acquisition, processing, storage, distribution, analysis, and interpretation. Okay, so genome sequencing, which uh, which study about the sequencing pattern of particular organisms genome, is very important because due to that we could understood the information stored in the form of genes. Okay, or we understood about the genetic diversity which is around us in the form of plants, microorganisms, in the form of animals, algae, fungi. There are different kinds of genes which are present, and they are producing different kind of products. Okay, so we are understanding our understanding about genetic diversity became more clear due to sequencing process. What is the role of NCBI? NCBI is in a simple form, it is a kind of library which stores information. Okay. How we store information, books in the library, the same way the NCBI stores information about different topics related with biology or chemistry or proteomics or genomics in the different sections. Okay. And whenever it is required for any scientist, they will provide them. The National Center for Biotechnology Information, that is NCBI, is a part of United States National Library of Medicine, that is NLM, a branch of the National Institute of Health. It is located in the Bethesda, Maryland, and was founded in 1988 through legislation sponsored by the Senator Claude Pepper. It houses a series of databases relevant to biotechnology and biomedicines and is an important resource for bioinformatics tools and services. Major databases include GeneBank for DNA sequences and PubMed and bibliographic databases for biomedical literature. Okay. So we are going to do one practical also in this particular uh, bioinformatics paper, and there is not only one practical. We are going to do two or three practicals about BLAS, FASTA, about the some practicals about proteomics, and they are really very interesting. You have to work not only in the laboratory, you have to work in the computer lab. Okay, how you even you can do it in the your mobile smartphone. Now next is the EMBL EBI. EMBL EBI is the home of the world's most comprehensive range of freely available molecular databases and resources. Resources help researchers to share and analyze data and perform complex queries in many different ways. Okay, so this is kind of another kind of database which is a molecular database, and this is freely available for anyone. You can go to this EMBL EBL and put your search, and whatever information is stored in that database you can easily access. Resources help researchers to share and analyze data and perform complex queries in many different ways. Okay. Next is OMIM, that is the Online Mendelian inheritance in man. Online Mendelian inheritance in man. You have to remember the uh, shortcuts and its explanation. Okay. So OMIM contains detailed full text referenced overviews of all human Mendelian disorders. That is more than 12,000 genes. Mendelian inheritance in man, that is MIM, is published in 12 prints editions since 1966. It is a compendium of information on genetic disorders and genes. It's a compendium of information on genetic disorders and genes. Okay, so whatever genetic disorders we have learned, you will get information about all these things and even about the genes. But this particular is only about the human being, not about other animals. Okay, so online Mendelian inheritance in man. It is comprehensive, then authoritative, and timely knowledge base of human genes and genetic disorders. So, whatever researchers are doing or whatever they are work they are doing in the laboratory, they submit it to OMIM if it is related with human genetics or human inheritance. And then OMIM stores it in the data format, and whenever it is required it will provide it to the other scientists working in the same field. It is compiled to support research and education in human genomics and the practice of clinical genetics started by Dr. Victor A. McCuskey as the definitive reference Mendelian inheritance in man. Now we have in the last chapter we have understood about the human genetics. There are so many different kind of human genetic disorders, and it is very difficult to understand sometimes why these genetic disorders are occurring, why these deformities are occurring in the developing fetus or in a progeny. Okay, so to understand the reasons behind these genetic disorders or the changes in the genes, OMIM is helping to the researchers. OMIM, that is www.ncbi.nlm.gov/OMIM, is now distributed electronically by National Center of Biotechnology Information, that's it, NCBI. It is integrated with the entrance suite of databases derived from the biomedical literature. OMIM is written and edited at Johns Hopkins University with input from scientists and physicians around the world. It is an authoritative full text overview of genes and genetic phenotypes that can be used by students, researchers, and clinicians. Okay, so again, one more database, and the next database, that is the PubMed. It's a bibliographic database of life sciences and biomedical topics covering practically all scientific journals in biochemistry and molecular biology. Okay, again, it is a database. Database is a the what you can say, the software uh, or a source which can store the data about different topics. So whatever is published in different journals, they will collect from the internet source and store it in the PubMed. It contains more than 20 million bibliographic records of biomedical publications. Just imagine 20 million bibliographic records they have stored about the biomedical publications, including free abstracts. More and more open access articles are freely available on the original journal websites in HTML format or downloadable as PDF file directly accessed via PubMed. It is one of the databases in NCBI and it contains about 20 million records of articles published from Medline and various other biomedical life sciences journals. Online books containing links to full text from PubMed Central and links to other NCBI molecular biology resources are also present in PubMed databases. PubMed offers different search operations, different search options according to the user's convenience, which is very easy to use even for a beginner. One can search articles by different searching methods. Search can be done by any query, word, by the name of the author, by the journal name, or by their PMID. Okay. PMID is PubMed identifier or PubMed unique identifier. Is a unique number assigned to each PubMed record. PubMed offers to sort the records according to the according to the date of published papers. Results and displayed year wise, which can be accessed from the graphical view displayed on the right side of the PubMed result page. PubMed also offers additional filter options to make our search more specific. Users can sort their search by choosing options from species, that is, human or other animals, sex, article types, languages, subjects, ages, and general categories. Okay, so we'll stop it here. In the next lecture, we are going to learn about the application of bioinformatics.